Hi, this is Dr. Jay Ganesh, working as an associate professor in SRM Institute of Science and Technology, Chennai, Katanguppam, Chennai. So today I will discuss about stem cell. Okay, so what is stem cell? What are the different types of stem cell? I will discuss. What is stem cell? So actually, stem cell is a specialized cells. They have a they have the remarkable potential to develop into many different cell types. So they have two important properties. One is they are able to make more cells like themselves. That is called self-renewal. The second important property is they can become other cells that, that do different things. That process is called differentiation. So that means they can become another type of cells via more specialized functions. So stem cells where they found it can be found in various locations including embryos, bone marrow, umbilical cord blood, specialized tissues. This is the pattern of stem cell division. So stem cell divide either into self-renewal cells or it can be differentiated into other cells with specialized functions. Okay. So other cells like it can be differentiated into muscle cells, nerve cells, bone cells, then blood cells. Okay. So these are called differentiation of cells. So this differentiation is mainly depends on the type of stem cell and the signal they receive. So based on the signal and the type only, it can be differentiated into other cells. Properties of stem cells. All stem cells have three general properties. One is capable of dividing and renewing themselves for long periods. Unspecialized, they can give rise to specialized cell types. So first one is capable of dividing and renewing themselves for long periods. So here the stem cells divide n number of times. That means the stem cells can divide indefinitely. So, for example, if we culture in the cells in the laboratory conditions for a few months, it can yield millions of cells. If the resulting cells continue to be unspecialized, like the parent stem cells, they are called capable of dividing and renewing themselves for long periods. So, normally cells undergoes 50 cell divisions and lose their self-renewal property. But in the case of stem cells, divides indefinitely by protecting telomerase enzyme activity. So the role of telomerase enzyme activity is that is responsible for that it is useful to prevent the shortening of the end of the chromosome during the cell division in order to circumvent the loss of important genetic material. That is the main role of telomerase enzyme activity. This is the pattern. The stem cell usually divides into either self-renewal cells or it can be differentiated into other cells. So now the another type of cell is unspecialized cells. So unspecialized cells mean they do not show any specific structures and does not having any specific functions. So examples for the specialized cells are this is unlike not some not like specialized cells. Okay. So for example for the specialized cells are one is red blood cells and heart muscle cells. So in the case of red blood cells, it has a specific functions. It can carry the oxygen through the bloodstream. So in the case of heart muscle cells, it can pump the blood to neighbor cells. So this example for specialized cells. So however, this kind of unspecialized stem cells can give rise to specialized cells, including heart muscle cells, blood cells, and nerve cells. So this unspecialized stem cell, pluripotent stem cells. So from the fertilized egg, after fertilization, it reaches the develop into blastocyst, then inside the blastocyst, cell mass will be there. We extract the cell mass, we can culture the stem cells. The third type is they can give rise to specialized cell types. So when unspecialized cell give rise to specialized cells, the process is called differentiation. So this is controlled by two signals. One is internal signal and another one is external signal. So internal signals are controlled by the cell's genes. So they are interspersed along with the strands of DNA and carry coded instructions for all the structures and functions of the cells. The external signals are includes chemicals which are secreted by other cells, physical contact with the neighboring cells, then certain molecules in the microenvironment. So these are the three different properties of stem cells. So this is the differences of stem cell into many specialized cells. So from morula, the next stage is blastocyte, then inner cell mass will be produced, then embryonic stem cell will be differentiated into other specialized cells like liver cells, pancreatic islet cells, intestinal cells, neurons, blood cells, muscle cells. So this process of differentiation of stem cells. You see about the potency of the stem cells, there are four types. One is pluripotent, totipotent, multipotent, induced pluripotent. So there are difference between the totipotent and the pluripotent. So totipotent means it can differentiate into all type of specialized cells in the body, including extra embryonic tissues. So that means the totipotent cell has the potential to divide until it creates an entire complete organism. But in the case of pluripotent, it can also differentiate into all cell types, but it cannot develop into the entire organisms. Multipotent means it can self-renew for a long period of time and can make multiple types of specialized cells. Another one is induced pluripotent. So this is artificially derived from the non-pluripotent cells by inducing the forced expression of certain genes. So totipotent. So here the totipotent stem cells can differentiate into embryonic and extra embryonic cell types, such as such cells can construct a complete viable organism. So these cells are produced from the fusion of an egg and sperm cells. The only totipotent cells are the fertilized egg and the cells produced by the first few divisions of the fertilized egg are also totipotent. Totipotent stem cells give rise to somatic stem, progenitor cells, and primitive germ-line stem cells. So this is the morula stage of embryo. So pluripotent stem cells are the descendant of totipotent cells. It can differentiate into nearly all cells. The cells derived from any of the three germ layers. So like endoderm, ectoderm, and mesoderm. So these pluripotent cells are characterized by self-renewal and the differentiation potential for all cell types of adult organisms. So these are true stem cells with the potential to make any differentiated cell in the body. Embryonic stem cell comes under this category. So this is a pluripotent stem cell differentiation. So it can produce any cell. So there are three germ layers like mesoderm, endoderm, ectoderm. So from the mesoderm, it can be differentiated into cardiac muscle cells, skeletal muscle cells, feeble cells, smooth muscle cells, red blood cells. Then from the endoderm, there is the internal layer. So it can be differentiated into lung cells, thyroid cells, pancreatic cells. Then the external layer is called ectoderm. It can be differentiated into skin cells of epidermis, neuronal cells, then pigment cells. So again, it is classified into multipotent cells. Stem cell can differentiate into number of cells. but only those of closely related familiar cells. So these are true stem cells but can only differentiate into limited number of cells. So examples are bone marrow contains multipotent stem cells that gives rise to all the cells of blood. Adult adipose tissue is another type of multipotent cells. It is a source of multipotent stem cells. So here the multipotent stem cell can be differentiated into myocytes, fibroblasts, astrocytes, stromal cells, adipocytes, fibroblasts, osteocytes. So oligopotent stem cell means it can differentiate only a few cells such as lymphoid and myeloid stem cells. So basic example is hematopoietic stem cells. So from the hematopoietic stem cell, it can be differentiated into NK cells, T cells, B cells, dendritic cells, platelets, erythrocytes, macrophages, neutrophils, eosinophils, basophils. So these cells are differentiated from the hematopoietic stem cells. Unipotent stem cell. So it can produce only one cell type. So they are own but have the property of self-renewal. Best example is muscle stem cell. So most epithelial tissues self-renew throughout adult life due to the presence of unipotent progenitor cells. Types of types of stem cells. One is embryonic stem cell, fetal stem cell, adult stem cell, induced pluripotent stem cells. So embryonic stem cell have the ability to form any fully differentiated cell of the body. So during the early stages of embryonic development, the cells remain relatively undifferentiated and possess the ability to become or differentiate into almost any tissue within the body. The embryonic stem cells derived from the early embryos. So what are the sources of embryonic stem cells are? One is embryo derived from the inner cell mass of the blastocyst human embryo. Sources are excess fertilized eggs from the in vitro fertilization or therapeutic one, SCNT method. So this is the blastocyte. Inside the blastocyte, embryonic stem cell will be there. So if you extract from the, if you cut the outer layer of the cells, we can extract the stem cells. Then we can culture under the laboratory conditions. So fetal stem cell means another type of stem cell or primitive cells types found in the organs of fetus. So it can be isolated from fetal blood or fetal bone marrow or any other fetal tissues, including liver and kidney. Adult stem cell means it is isolated from the any tissue or organ of an animal or human. So adult stem cell can divide or self-renew independently. So it is generally thought that adult stem cells are limited in their ability to differentiate based on their tissue of origin. Sources of adult stem cells are bone marrow, liver, retina, skeletal muscle, intestine, brain, dental pulp. Induced pluripotent stem cells. So induced pluripotent stem cells are created by inducing the specialized cells to express genes that are normally present in embryonic stem cells and that control cell functions. So induced pluripotent stem cells are useful tool for drug development, modeling of disease, and the scientific hope to use them in transplantation medicine.